Hey everyone, this is Dual Credit Biology 1, Unit 2, Lecture 5. Today we're going to be talking about lipids and nucleic acids. So we've discussed basic chemistry, we've gone into organic chemistry, we've discussed hydrocarbons, functional groups, macromolecules, and then we also discussed carbohydrates, which were one of the four macromolecules. Today we're going to tackle the second and third type of macromolecule, lipid, nucleic acid. So go ahead and grab your PowerPoint so you can follow along and make notes. I'm going to share my screen now so that we can kind of follow along and talk a little bit about it. Remember, as you're watching these PowerPoints and everything, I'm hitting the highlights, things that I think will help you to understand some stuff from your book, but it's very important that you be proactive and that you read your textbook and that you write down any questions that you may have of things that you don't quite understand, so that later when we have a Zoom session as a review, we can cover any topics that may be giving you some trouble. So let's start with lipids today. So these are things that you've talked about before in other science classes, but lipids are a very diverse set of molecules. And of course, they're hydrophobic, which means they hate water. They are also made of carbon, hydrogen, and oxygen, very similar to carbohydrates, but the difference is there's no general formula. We learned before that carbohydrates have a CH2O. There's a 1-to-1 ratio with carbon and oxygen. Lipids aren't like that. There is no set formula. We do have a carbon-oxygen ratio that is very high in carbon, meaning there's more carbons than there are oxygens. We are looking at lipids being made of two different kinds of molecules that are smaller. They consist of fatty acids and then also something called a glycerol, and that OL tells us this is an alcohol. And then of course we know OL or alcohol means that there's going to be some hydroxyl functional groups. So a fatty acid is just basically a really long carbon chain, anywhere between 12 to 18 carbons long. And then at the end of it, you're going to see a COOH. So that's how you kind of have an idea that it's, because that's a carboxylic acid. So that tells you it's an acid. And then on the other end, you're going to find a methyl group. And remember, methyl is what makes it hydrophobic. That's going to make it the fatty part on the other side. So that's how we get a fatty acid. So when you're looking at it, it looks like this. So if we were going to make a lipid, join this glycerol to this fatty acid, we would pull off our water through dehydration synthesis, join them together, and the type of bond that forms in a lipid is called an ester linkage. And then there we would have our fatty acid with the glycerol. This is called a triglyceride because there are 3 of these fatty acids attached to this glycerol. And some of you have heard of triglycerides. We'll talk about them a little bit more in just a few minutes. So moving on. So triacyl glycerols, these are 3 fatty acids that are joined to one glycerol by that ester linkage like I just showed you in the last picture. Of course, the carboxylic part, the carboxyl group that we see at the end is what makes it the acid, and then the OH at the other, the OH is what makes the glycerol an alcohol, and then of course the methyl, the CH3, is what makes it the fat. So when we talk about fats, there's a lot of discussion and just layman's discussions, just everyday discussion about fats. You've probably heard people talk about saturated fats, unsaturated fats. You may have even heard of something called trans fats. A saturated fat is basically a fat that when you look at it, it is completely saturated with all the carbons. So let me just kind of draw right here. If we were talking, and this is a carbon skeleton, this is not an actual drawing of a fat, okay? I'm just showing you as a hydrocarbon skeleton. If we were talking about a saturated fat, if you'll notice, this carbon skeleton would be indicative of saturation in hydrogens, meaning that everywhere there's a possible possibility for a hydrogen to join, it would join. And notice there are no double bonds. It's all single bonds. This is what a saturated fat would be. There's no double bonds, okay? Now, an unsaturated fat's a little bit different. An unsaturated fat does have multiple bonds. So you end up with something like this. In this case, all the bonds all the bonds are taken, but notice you don't have the maximum number of hydrogens that you could normally have if it was single bonds. Meaning that there could be more hydrogens attached, but because of this double bond here in the middle, it messes that up for us. So double bonds, they tend to cause kinks in the molecule shape, meaning they don't lay down properly. And because of that, it makes these particular types of fats different states of matter at room temperature. Fats tend to be solid at room temperature, like butter. Oils like canola oil or vegetable oil or peanut oil, those tend to be a liquid at room temperature. And the reason why is because of those double bonds. Fats are usually solid at room temperature because there are no double bonds. Everything lays nice and neat. I am a fanatic for Tetris. I can play Tetris for hours upon hours upon hours. And Tetris, if you don't know what it is, it's a block stacking game where you just basically are given different combinations of 4 blocks in these different shapes, and your job is to try to line them up perfectly, and then when you get them perfect, your line disappears. I'm really good at Tetris, really good. I love to pack the car when we go on vacation because I love for everything to fit just perfectly. The whole family knows that that's part of my vacation fun, is packing the car, because I have an eye for that. I can look at our luggage, I can look at the stuff that we're taking, I can look at the back of the car, and in my mind, I know how I need to put it so that I can get the maximum amount of space from that vehicle and everything fits nice and perfectly. Fats do that because they are saturated. There are no double bonds, there are no kinks, everything lays perfectly, it stacks perfectly. And so therefore, because it stacks perfectly, it causes that melting point to be higher, and so it does not melt at room temperature. Now with oils, because there's a double bond, it makes it kink. So it's kind of like, you have 10 suitcases and 9 of the suitcases are perfect rectangles, and then you got that one kid that shows up with this duffel bag that's all lumpy and bumpy. That lumpy bumpy duffel bag is going to throw off your nice neat structure in the back of your car, just like an unsaturated fat will. And so those double bonds cause that kink, so they don't lay nice and neat, and it lowers the melting point, and that's what makes it a liquid at room temperature. So here just kind of gives you a little example. So these are your saturated fats, and you can see how they're solid at room temperature because they kind of lay nice and neat. And right here, this double bond right here in the middle causes it to kink up, and then that leads to a change in the melting point, which of course leads to an oil. Fats differ in which fatty acid is used. Some fats are used for energy storage. Some fats are used for other purposes like insulation, things like that. In general, fats are energy storage, cushion, and insulation, but specific fats do specific things within the cell. One question that I always like to ask people, we know that fat is an energy source and starch is an energy source. So which one gives us more energy? And it's actually a fat. Fats have more carbon-hydrogen bonds per mass, which means that you get more energy from fats. That's why if you go to the gym, they'll have like this poster up usually somewhere in the gym that talks about your fat burning zone. When you get to a certain point, you've burned up all your carbs and you begin to burn fat for energy, and that's how you begin to lose weight or lose inches. It's because you're getting rid of that fat that's just hanging around, because your body begins to pull that for energy rather than other things. So a phospholipid is a specific type of fat that we see. Their difference is it only has 2 fatty acids in comparison to 3. Um, the third hydroxyl group or functional group is joined to a phosphate containing molecule, hence the name phospholipid. Phospholipids are really important. Looking at this picture right here especially, you probably recognize this from Biology 1. That's the little chemical or little fat that we use to make our cell membrane. The cell membrane is a phospholipid bilayer. Later, we'll talk about what makes this or why this is important, this little kicked leg right here. But the reason why it kicks is because notice there's a double bond, and we know double bonds cause it to kink up. And but that's important in the cell membrane and how it behaves. But this is a phospholipid. It has a hydrophilic head, meaning it loves water, hydrophobic tails, which means it hates water. So of course they spin themselves around to protect the legs from the water inside and outside of the cell. And then the hydrophilic head is what faces the water outside of the cell or inside of the cell. So that's really important because it self-assembles into these bilayers, and that bilayer is incredibly important to the function of the cell. When your cell membrane doesn't function properly, the cell doesn't function properly, which can cause all kinds of problems with the organism. We have to have our cells functioning like they're supposed to, and in order for that to work, we need that structure of the cell membrane to work. So once again, this is a perfect example of how things build in biology, how function depends on structure, even down to the atomic level. Because if the atoms aren't right, then your molecule's not right. If your molecule's not right, then the cell can't function like it's supposed to. And so I think that's a really important point to drive home, is that structure and function are very dependent on one another. A steroid is also a lipid. We hear a lot about steroids, but you got to remember there's lots of different kinds of steroids. We hear the word steroid, everybody thinks bodybuilders and, injecting steroids and getting all buff. But, there's steroids are used for a variety of things. It's a lipid, but it's typically rings that are fused together, and the way that they differ is based on what functional groups they're. A good example of a steroid that I showed you a couple of lectures back was the lions, the female and the male lion. There was a difference in the functional groups that were attached to the rings, and so it created a different sex hormone. Sex hormones are considered steroid or are steroids. Cholesterol is a steroid, and it's very important. We think cholesterol is bad, but we have good cholesterol and we have bad cholesterol. We want to keep our bad cholesterol low, but our good cholesterol high, because cholesterol is very important to our cells. So that kind of wraps up steroids. A lot of this information, or that wraps up lipids, a lot of this information we're going to talk about a little more in depth as we get to different units. So we'll talk more about like phospholipids and steroids and even carbohydrates when we get to like the cell and the cell structure. And then when we get to different functions of the cell, we'll talk about some of these macromolecules a little more in depth. But for right now, we're just kind of hitting the highlights based on their structure. So let's take real quickly a minute and go through nucleic acids. Nucleic acids, I don't expect to take very long because it seems like this is the one thing that really gets hit hard in biology. So a nucleic acid is a polymer. It's put together of monomers, but it's what we call an informational polymer. And the word informational to me, it's like a blueprint. Its job is to tell us how to put you together. Of course, it's made of carbon, hydrogen, and oxygen, but also we have nitrogen, and there's some phosphorus in there as well. There is no general formula because everybody's nucleic acids are different. Your nucleic acid will be different from my nucleic acid because our DNA is different. These are DNA and RNA molecules are made up of this macromolecule. So nucleic acids are polymers of monomers. The monomer for a nucleic acid is known as a nucleotide. That's very important. Nucleotides have 3 parts. You're going to have a nitrogenous base, like adenine, cytosine, guanine, thymine, or if you're talking about RNA, uracil. And then you're going to have a sugar. It's a pentose sugar, which means it's 5 carbons. There's ribose, and then there's deoxyribose. And then you're also going to have phosphate in there as well. So a nucleotide has a nitrogenous base, a pentose sugar, and a phosphate. So I always tell people sugar, phosphate, base, sugar, phosphate, base. So when you look at the nitrogenous base, your bases are carbon and nitrogen rings, and they are put together either as a single ring, which is known as a pyrimidine, or a double ring, which is known as a purine. Later on, when we talk about how DNA was discovered, its structure discovered, we'll talk about why that's so important, because the shape of DNA hinges on that particular building of nitrogenous bases. But those bases attach to and form the rungs, the steps of a ladder. The pentose sugar, of course, is 5 carbon. We said ribose is an RNA, deoxyribose is DNA. The only difference is there's a hydroxyl group that's different on the 3rd carbon. And the DNA is a deoxyribose, so it's lost that oxygen, and that's why it's different than the ribose. So here's examples of what it kind of looks like. These are the nitrogen bases I was talking about. You can see that they're rings of carbons. These are our pyrimidines. They're the single rings. And these are our purines, which are the double rings. There's our little deoxyribose. See, it's missing that hydroxyl group right there. And then there's our ribose, and it has it. And then these get put together. So this is your phosphate groups, are these little circles. This is your pentose sugar. And then there are your bases. So it's a sugar, phosphate, and a base that all get put together. So a phosphate, there's your sugar, and there's your base. And then, of course, we know that the sequence of the bases is what makes each individual a little bit different. All right. So DNA, deoxyribonucleic acid, it's what makes up our genes. It is the genetic information source for most life, not all life, but most life. It has a very specific structure, which we will get to very in depth later on, but we know it as a double helix. RNA is a little more simplistic, but it is not by any means not necessary. It is what we use for structure and protein synthesis. It's very, very, very important. It's also the genetic information for some viruses. We talk about a lot of our viruses later on. We will learn that they are RNA viruses. And so that wraps up nucleic acids. I'm going to stop share right here. So I would go back and I would read through my chapter to this point. Make sure that you know your notes, that you've studied your functional groups, that you go back and look at the basic chemistry. We have one more lecture on proteins, which is kind of a lengthy lecture, and that will be it for this unit. So if you have any questions, feel free to email me, and I will see you later.